Welcome to botany class. Myself M. Manaja, lecturer in botany, Government Degree College in Guntagal. Topic is embryo sac types in plant embryology. Learning objectives. To understand the scope and importance of embryology, to acquire the knowledge on structure and development in megasporangium, to differentiate between microsporogenesis and megasporogenesis, to identify and illustrate the types of female gametophyte, that is, embryo sacs types, to know fertilization, endosperm, and embryogeny. Embryo sac. What is embryo sac? Embryo sac is the female gametophyte. It is also called megagametophyte. Megagametophyte produces an egg cell for fertilization. A mature embryo sac has an egg apparatus at the micropylar end and three antipodal cells at the chalagal end with two polar nuclei at the center. This is the structure of embryo sac. Embryo sac originates from megaspore mother cell. The process is called megasporogenesis. The megaspore is the first cell of the female gametophytic generation. Embryo sac development is based upon the involvement of nuclei take part in the formation of embryo sac. There are three types of embryo sac development are there. First one monosporic, bisporic and tetrasporic. They have been further divided. Monosporic having two types, polygonum type and enothera type. In bisporic, having identified in only one type of, that is allium type. And here tetrasporic embryo sac development having peperomia type, pinia type, drusa type, plumbago type, plumbagella type and adaxa type. This chart explains the development of different types of embryo sacs in angiosperms. Here we see that the differences between monosporic and bisporic and tetrasporic. Monosporic of megaspore mother cell divides mitotically and gives the tetrad cells. In this tetrad, only one cell is actively participate in the formation of embryo sac. Remaining three are degenerate. And bisporic megaspore mother cell divides, first meiotic division gives two diad cells of the megaspore. In this stage, the two diod cells are involving the formation of embryo sac. And tetrasporic. The tetrasporic megaspore cell undergoes division, gives tetrads of megaspore. In this tetrad of megaspore, the four nuclei of the tetrads involving the formation of megasporogenesis, that is embryo sac structure. These are the three differences between them. Let us see the first type of embryo sac type development. Embryosac, monosporic embryo sac type. The formation of embryo sac from a single megaspore cell is called monosporic development. Based on number, nucleus at the maturity, monosporic again divided into two types. First one is polygonum type. It is having 8 nuclei. Second one is Enothera type having 4 nuclei. Let us see the example of polygonum type development. Polygonum type of development is first described by the Strasburger in Polygonum divaricatum. This is also known as a normal type. It is found in majority of angiosperms. In this polygonum type development, the chalagal megaspore of tetrad is take part in the formation of embryo sac. The functional megaspore nucleus enlarged divides into their respective poles. Each nucleus divides twice, forming 4 nuclei at the pole, resulting 8 nucleates. At the micropolar end, 3 of the nuclei are organized into egg apparatus. 2 are central nuclei. At the chalagal end, 3 are antipodals. The two polar nuclei coming to fuse together at the center forms secondary nucleus. The center of the embryo sac is diploid and all other the haploid. The arrangement of 8 nuclei in polygonum type embryo sac. The 8 nucleates are arranged like 3 plus 2 plus 3. That means 3 are the egg apparatus, 2 secondary nucleus, 3 antipodal cells. Next, bisporic embryo sac type development. The megaspore mother cell forms 2 diod cells after first division of meiosis. The 2 diod cells are involving the formation of bisporic embryo sac formation. So, it is called bisporic embryo sac development. This type of embryo sac development was described by Strasburger in 1879. It is identified in the plant name Allium fistulosum. In the diode cells, upper one is degenerates, but the lower one develops into the embryo sac and its nucleus divides into 8 nuclei. The arrangement of 8 nuclei also having similar to the polygonum type, that is 3 plus 2 plus 3. This type of development also found in families like Liliaceae, Orchidaceae, Podostomaceae. Here we observe that a process of bisporic development. Megaspore mother cell gives first meiotic division, gives diode cells. First, one is degenerate, only one is functional. It gives meiosis II division and also further divides into mitotic divisions, resulting finally 8 nucleate embryo sac structure. One is a egg cell, other two than synergids. They are lying at the micropylar end and chalazal end. Three are the antipodal cells at the center, secondary nucleus. Next one, tetrasporic embryo sac development. In the megaspore mother cell, after meiosis of all 4 haploid megaspore nuclei take part in the formation of the embryo sac structure. They are not delimited by the wall formation in tetrad megaspore. In this type of formation, some are having 16 nucleate embryo sacs are formed and in others only 8 nucleate. Depending upon the organization of arrangement, they are following arrangements are there. One is 3 plus 1 arrangement. It is identified in Fritillaria, Plumbagella and Drusa types of plants. Second type of arrangement having the 2 plus 2 arrangement. Example in Adoxa plant. Third one is 1 plus 1, 1 plus 1 arrangement. Example in Peperomia type and Pinia type of plants having these type of arrangement. Let us see the development of Peperomia type of embryo sac structure formation. Example of tetrasporic development. The Peperomia type of embryo sac having the nuclei arrangement 2 plus 8 plus 6. The Peperomia type of development was reported by Campbell and Johnson in Peperomia pellucida. After meiosis, each of the 4 megaspore nuclei divides twice, forming 16 nuclei which are uniformly distributed at the periphery of the embryo sac. The 2 nuclei lying in the micropylar part get organized into an egg cell and synergid. 8 nuclei come to fuse in the central region of the secondary to form secondary nucleus. That is 8N. That means 8 nucleate. The remaining 6 nuclei form antipodal cells at the periphery of the embryo sac of the chalajal end. These are the antipodal cells. This is the development of peperomia type of embryo sac formation. Now, recall the topic. Megaspore undergoes division, forms embryo sac. There are three types of embryo sacs or development is present in the plant. Monosporic embryo sac develops from a single megaspore. It is identified in polygonum type. Second one, bisporic embryo sac develops from megaspore mother cell of dyad cell. Example, allium type. Tetrasporic embryo sac develops from 4 haploid megaspore nuclei. Example in peperomia type. Thank you.